So basically, I'm going to show you guys use BCFtools. Feel free to follow along. Hopefully you've installed it. It's a really powerful tool. I think I pretty much use it almost every day. And essentially what it is, it's a program written in C. So it's extremely fast, and it's designed to work with VCF files. So as a reminder, a VCF file just contains variants, right? So in this directory, I have a VCF file and its index. Now, for most, not all the functions of BCFtools, but for most of the functions of BCFtools, it does need to be bgzipped, which here you can tell by the .gz extension, and it must also be indexed using tabix. So the basic overview is that you do like a bgzip. Your file has to be sorted, so you can actually use a BCFtools sort to help sort your file. And then after you sort it, then you can do a bgzip on the VCF, and then once you do that, then you do a tabix of the VCF.gz, and then you'll get this index file. And that's basically like a small introduction of that, right? So I have this file. We'll look at it. But you can tell from the description that it's whole genome sequencing. You can tell by the date; it's kind of old. Yeah, it's really old. And but it's SVs. So these are structural variants. So these are like deletions, insertions, duplications, things like that, and they have genotypes. So let's take a look at this file. So actually, really, with BCFtools, you can just look at it, right? Doesn't matter what format it's in. Your BCFtools view, and it will produce a readable file for you. Okay, so you can see it has like all the header information. It is a structural variant file. It has structural variants in it, so it's slightly different. You can see that it has chromosome position, the name of the variant. Sometimes it'll actually have the alleles. Here it's showing a deletion of basically these nucleotides. Pretty small deletion. And then here it has a lot of information that you can use in the info column. And then here you have your samples. And there's a lot. There's a lot of samples in this one. So just to show you how many samples there are, you can see right here that it's quite a bit. Now, what's also interesting is that it only has genotypes. And here you can see, maybe some of you are familiar with this, that it has this pipe character. Basically, all this means is that it's phased. I'm not going to talk about this in detail right now, but you can appreciate that there's a difference between 1 pipe 0 and 0 pipe 1. So, yeah, I wrote some README information in these, and we'll just kind of go down the list, and I'll show you the utility of BCFtools view. So, like I said, you could just open it and look at it. Now, you can also output in bgzip format. So in this case, it's a little circular, right? Because you're like you're opening a .gz and then you're outputting the .gz. So, but what's good is that you can actually apply this restriction. So now let's restrict it to just one sample, and then you can also tag along this option. So let's show let's show this in action. So now you'll notice that instead of having those 2,000 samples, we just have one sample, and that's really useful. You can also chain together. I think 096, 110. You can chain like more than one sample, and now you can see there's 2 in there, right? Also, you can have a file, and if the file has samples 1 per line, then you can just use that instead. So here, instead of using the lowercase s, you use the uppercase s, and then the sample file, and it'll basically do the same thing. See that? Okay. So yes, so yeah, we basically covered these. Now, you might use it a lot to subset a VCF to a specific region, right? So doing this, you'll use the R option for region. Now, it can be as broad as a chromosome or 2 chromosomes, or you can give a range of positions like this. And you can keep chaining it and mixing it all up. So here, let's actually just look at something like this. So now you can see that there's not that many variants in here because we only restricted it to 1 megabase of the genome. And when it will select the variant, it will only look at this position. It will say if the start position of the variant is within within the regions that you defined. For structural variants, as you can tell, there's an end position, right? Because with an SV, you have the beginning of the deletion and the end of the deletion, right? For example, and this information, the end, it's in here somewhere. Here it is. So you can see that there's the end position, right? So there's not really a clear way to include the end position too, right? Because what if you have a structural variant where the end position is within your range but not the start, right? This is a limitation of BCFtools and the VCF format. And there's actually been a lot of debate within the structural variant community about how to correctly address for this. Obviously, the easiest thing to do is just to add an end position here. And I don't know why it's been almost a decade and people haven't done that yet. Anyway, so yeah, that's basically kind of like basic subsetting to a region. Now, you may have a situation where you want to subset to a file of regions, right? Say for example, you have positions of genes that you're interested in, or you have a position of known risk variants, right? Then you can put those genomic regions in a bed file or in a similar file that's formatted like a bed, but with a 1 base position. Basically, the rule of thumb is that if you're going to use a bed file for BCFtools view, you need to use the .bed extension. It can also take bed.gz. But if it does not have the .bed extension, it will assume it's in 1 base position. And if you're a little confused about the differences between 0 or 1 base, I believe on the homepage of the Gleason GitHub page, there's actually a link that discusses that. And if you have trouble finding it, you can just let me know and I'll point it out. Okay. So yeah, what happens here? So let's take a look at this. And please stop me if this is like, if I'm going too fast or anything. So you can see here in this bed file, I have a region which are the first 10 million base pairs on chromosome 1. And if I run this, then you can see that indeed we get everything that's from between that position, right? I mean, obviously you will have like many positions, right, in a file, and that's why you would want to use this utility. Okay. Now, I really highly recommend that if you feel like you really want to get a good grasp on BCFtools and subsetting, sorry, my cat is chewing on something, subsetting, filtering data and kind of like being like a power user, then take the time to look at this link because there's tons of different expressions or conditions that you can use to help filter your data. Such are, say for example, you only want to get the variants that have pass in the filter column. That's a very common thing to do, right? So to do this, you use the I flag. Now the I option tells you include. So essentially in natural language, you're saying, open the file, BCFtools view, let me look at it, and include all the variants that have a pass in the filter column, or that it's equal to pass, right? Yeah, and so now you can also parse out information within the info column. And this is what I was getting at with like the SV type or the end position. There's also a lot of other information in the info column that you may want to use in like SNP data or other things too. So essentially, in order to do this, you specify the column. So here, info. But then if you have like a subheading within the column, right, like SV type or end, you give it a /, and then you just, you basically do it like this. So here, let's take a look, right? Now you can see that basically they're all deletions. And you can take my word for it because one, you can see that there are a lot of pin-del calls. The alt allele here is a loss of a copy, CN0. And yeah, if you just keep looking at the end, you can see SV type del, SV type del, SV type del, right? So that's actually like a really cool thing to do, right? Because, when I was younger and dumb, I used to write like scripts in order to pull out this information, to like really make sure that like it's equal to del or something like that. But you can just do this in the BCFtools, right? And you can save it into a new file, right? And you can do anything you want with it, right? Now, perhaps you're not aware of this, but sometimes you don't have to have an equal statement, but you can have a match statement, right? So if you give the tilde, it will match del as a string. So sometimes, for example, you'll see something like del alu, or maybe someone had a mistake and did this or something. Now if you do this, then now you actually get. Let's actually see if I can find one. Yeah, it actually found one somewhere. But basically, I'm not gonna like sit here and like look for it if I can't, spot it with my eye right now. But basically, there are instances where the SV type in this list is not. It's like del underscore something. Yeah, right here you can see, right? So here, SV type del alu. So if you did the equals, right, you would miss this. But if you by giving the tilde, you're able to match it. Is that clear? Okay. So it's really powerful, right? Okay. So, say for example, another common filter that people do is that you want to be extremely stringent when it comes to the genotypes, right? You don't want any case or instance of a missing genotype. And missing genotypes in VCFs are typically encoded as a period, right? So as I mentioned, this is a matching operator. It's different from the equals, right? And you can essentially, apply the E flag, which is exclude. Now, the thing is that if you start playing around with this, you'll notice that you cannot have an exclude. You cannot have two excludes. I think you can chain things with like the AND operator and things like that. I believe all that information is in that link that I sent. So yeah. So here, let's take a look at it. And actually, yeah, I think that's fine. All right, it's not like, yeah, then you have to give this like single quote, right? There it goes. Now if like, if you want to look, I mean, there's just not any periods in there. right? We have like good genotypes, right? Okay. So I think that basically covers. Oh, and then sometimes, like another thing is like, maybe you want to get a situation where only one person has the variant and everyone else is 0, 0. So you can use allele count equals to 1, and it'll do something like that, right? Basically, just anything in here, right? You can just take a look at it. It's pretty thorough, and it covers a lot of information, so that instead of writing a script that's taking like maybe 10, 15 minutes, 30 minutes of your time, you can just do a one-liner command, make an output file, and then parse that output file as you need it. Hopefully that makes sense. Okay, so query. I use query quite a bit, and maybe query is not the correct name for the program. I think it should maybe be called like reformat or format. That's essentially all you're doing. You're essentially taking the BCF and you're reformatting it into a text file for whatever you want to use it for. Now, I typically use this with respect to structural variants when I want to convert it to a bed file. And the reason why I do this is, as you can tell, is that you need the end position to do certain analyses. Like you can't just rely on the start. Sometimes you need to know the end position. So the way to do this is that you must give this format string, right? And this format string, the header information is typically prefixed by a % sign. And you can give any delimiter that you like. Personally, I like tabs, and for a bed file, it has to be a tab delimited file. So here I'm giving the tab character. Now, one thing you may not pick up is that BCFtools is very nice in the sense that they have offered a zero-based position, because bed files are in zero, by definition that they have to be a zero-based file. So this pos 0 is essentially the pos -1. That's all it is. Okay. Similar to before, to get the information in the info column, you just you give the info header / the subheader, subbing, in this case end, and here SV type, and as you can tell here, you must provide the new line character. If you don't, it'll just print it all in one big line, and that's not fun for anybody. Now, obviously, a lot of the same filters in Vue can be applied here. So let's take this. Let's look at this for an example. So now you can see it's in bed format, right? Chromosome, start, and SV type. Now, as you can appreciate, some of these ends have a period. This means that they're missing, that the SV end position was not in the VCF. That's okay, because in this case, what we're looking at is an insertion. Insertions technically don't have an end position because you're taking a piece of DNA, right? And you're inserting it into a region of the genome, right? Like kind of like a particular place in the genome, right? So there's not really like you can't say like, oh, there's a deletion from position 1 to position 10, right? You'll say there's an insertion at position 5. Well, say for example, I don't want any of these insertions. Say for example, I want only duplications, right? So I'm going to say include info, and in this, so there's a slight difference, right? Like when you give the format string, you do have to give the % signs, but when you give the info or the include or the exclude options, you don't give them. Maybe they'll work with them. I haven't tested it, to be honest. But anyway, info SV type matches del, and there you see. Now we have all the deletions, and it goes all the way down to the end of the file, right? So that's really awesome, right? Because now I have a file that contains that I can actually use to intersect genes and other things too, right? It's So quick conversion, right, of VCF to a bet, just in one line. I used to write scripts to do this. Yeah. Okay. Now you may ask, okay, what if I want to get the genotypes for the samples? So what's really useful about query is that if you put the information in brackets, as you see right here, so in the first line there are no brackets. So when there are no brackets, you're essentially doing the query operation on every variant. Okay. When you do it, when you have the brackets, you're now applying the operation for every sample, right? So if you have 3 samples and 100 variants, this command will produce 300 lines. Here you would have 100 lines. Hopefully that makes sense. So basically, when you use the brackets, there are some special variables that you can use. One is sample. Sample is just the name of the sample. And then anything else that's in the format column. In our case, there's only genotype in the format column. Perhaps you've seen other information like DP, which is the depth, or AD, which is the depth with respect to the alleles. You can chain those too, right? You could just, and then you can also mix like, you can mix different delimiters, right? They don't necessarily have to be just tabs. I mean, I like tabs, but say for example, you want the last column to be a mix of commas and and tabs, or maybe you want to use a colon here, right? Or maybe you just want to say something like, hi, it'll work. Why not? I won't save it here, but yeah, let's look at this one. So, I basically have this command, and I can't do that, it's on this computer. And then, I can just space, this is a test. And it's just like it's a new line. I think that will work. I don't know. Yeah, see, now it works, right? And so you can see right now that essentially, if I can, yeah. So basically you can see that it's printing all the samples with respect for the variants. And here it has the genotype and anything else you want to put on there, right? So it's a super powerful tool, right? Say that you only want to get genotypes for NA12878 and only variants where she has an alt allele. So essentially it's the same format string, but now I'm just adding some of the previous options that we've used before, right? So genotype is equal to alt, or using the include option, and then using the same sample option. So, yeah, why don't we just run this one and take a look at it. So you can see that basically it's printing out all the variants for this individual where she has a mutation, right? And as this file says, you can get alt homozygous variants, and this is in the this is actually in the expressions link I sent in the other file too, where AA is like alt, or you could be more specific and give something like this. And the last thing I'll give you is that what's really nice about BCFtools Query is that you can also just print all the samples. So sometimes you're just like, I just want to list the samples in the BCF, and this function will do it for you. I think maybe a student showed me this, to be honest. And right there, it just prints all the samples, and you can like count them. Oh, I have 2,500 of them in there. So so yeah, that's basically it. That's kind of like a whirlwind tour of BCFtools. Highly recommend getting familiar with it, especially if you are considering like working with BCF files for in genomics and stuff. It's not going to go anywhere. Like people are going to use BCFs for a while. So yeah. So with that, I'm just going to think I'm going to stop sharing, and then I'm going to stop